We'll wait for a couple more minutes to join, other people to join because nobody else has joined. 2, 3 minutes, we'll wait. Okay, I think now we can start. Okay, so welcome to week 4 of Advances in Omics. Uh, Last week we discussed about a few different sequencing technologies. Last couple of weeks. Today we'll be more moving into more of the applications of the data that we find from this sequencing technologies. How do we use this data to infer uh, evolutionary insights into different organisms, different species? Uh, I think last week also we talked about genome assembly and a little bit of variant calling and these things. So the topics that I'm going to cover today and that has been covered in this week are basically downstream to the topics that were covered in the past 2 weeks, where we learned about sequencing technologies. Now we are going to see what use we can make of the data from a evolutionary perspective, particularly for this week. Okay, so let's get started. So first comes the concept of pseudogenes, right? So these are, as the name suggests, these are non-functional DNA sequences. They look like genes, but they're not actually genes. They resemble functional genes, but they have lost the ability to code for proteins. So in some evolutionary history, these genes were active. These genes used to code for proteins and amino acids. But due to evolution, due to selection, these genes have lost their ability to code for functional proteins. So they serve as molecular fossil records for evolutionary history. As in, like you can identify these pseudogenes and you can make an evolutionary claim that why this inactivation has happened and what kind of selections has these genes gone through. Right? So, what are the reasons for pseudogenization? So, basically, conversion of a protein coding gene to a pseudogene. Right. So, firstly, can be missing promoter. It can be just the promoter got mutated of that gene, or the promoter got deleted or knocked out of the DNA. So, although the gene is present, since the promoter region is not present in the gene, that the DNA polymerase cannot come and bind and replicate the DNA of that region, or use transcription to convert that portion of the DNA to mRNA. So that's a pretty interesting way to lose a function. You just lose a certain part of the gene, which is a very important part, which is actually upstream of the gene, so that that gene does not has lost the ability to form mRNA. Missing start codon. So this is something that we see very frequently. The start codon is very specific, right? It codes for methionine, the AUG codon. It can just one mutation of the start codon can actually hamper the whole gene. And if you miss out the start codon, and if there are no AUG sequences in the whole gene apart from the start codon, so that your gene mm, transcription translation initiation will not happen, right? Then again, frameshift mutations. These are like different types of mutations that can happen in different portions of the gene that can be affected, right? Premature stop codons, which we call as stop gain mutations. So changing of a DNA base pair, which actually codes for a termination, right? Then you have missing introns or partial gene deletions as well. So one good example of this are the olfactory receptor genes in humans. So if you look at human genetic data across different populations, you'll find a lot of loss of function variations in these olfactory receptors, which are very common in the population, which technically should not be, because these are genes that code for different olfactory receptors, different olfactory chemical molecules that help us uh, smell. But yeah, I think this picture has come down. Sorry for that. Yeah. But again, so millions of years ago, when humans were not technically humans, they possess these genes and they possess these receptors and for which was used at that time for living. But again, we have come down such a long way after evolution that we do not need these genes anymore. So almost 50% of olfactory receptor genes are pseudogenes in humans, compared to dogs and mice. If you see their proportions, if you see around, so the blue part here is the pseudogene and the red part is the functional gene. So if you compare dogs with humans, below you see a greater proportion of pseudogenes in humans, right? So because we have through natural selection, we have lost certain of these characteristics to smell certain to have a strong sense or strong olfactory receptors compared to uh, species like dogs. Right. So the evolutionary implication was that we, as primates, we had reduced reliance on smell compared to this kind of species like dogs. Okay. So one way through which so this was the concept of pseudogenes, but the idea is that you have a functional gene at a certain time in your evolutionary history, but that gene has lost its ability to perform as a gene in the current era. So one way to achieve that is basically whole genome duplication. Now we are not talking about a single gene, but the whole genome can be duplicated, right? So these whole genome duplication events have shaped the history of evolutionary lineages. One such duplication, this is one example of it, has been implicated in a particular type of fish called teleost fish, and which is basically the most species-rich vertebrate plant. After initial controversy, now there is solid evidence that such events took place in common ancestor of extant teleosts. It is termed as teloid-specific WGD, which is whole genome duplication. Uh, After the whole genome duplication, the predicted whole genome duplication was around sometime around like 80 to 300 million years ago, where the whole genome got duplicated, and after that, certain genes had certain fates, right? So the duplicate genes were mainly non-functional because of lack of selective constraint preserving both. So there is no point in having a same gene encoding for the same protein in multiple copies, right? So it's the same gene. You do not need that. So for during by selective mechanisms, selective constraint, uh, it turned out one copy of the gene turned out to be non-functional. So. This is another very good example of this is like it's not exactly the same as whole genome duplication, but if you think about X-linked inactivation in women or in females, in humans, where females have two copies of the X chromosome, where males have one copy of X and Y. So there to control for the dosage of the gene expression in X chromosomes of human females, one copy of the X chromosome is silenced. So that is what is known as X-linked inactivation. So that is also an evolutionary mechanism to do this. So here also similarly, since you had multiple copies of the same gene, so it can one copy can be the extra copies can be non-functional due to selective constraint, right? Mechanisms that act on preservation of duplicates are subfunctional, partitioning, sorry, partitioning of ancestral gene functions on duplicates, neofunctionalization, which is basically assigning a novel function to one of the duplicates. That is also very important. That is how like further selection on these kind of genes will occur. And dosage selection, which is basically preserving genes to maintain dosage between the interconnected components. As I said, some a good example of it would be X-linked inactivation. Now, uh, so all this information in terms of evolutionary insights from humans and different species, but also we want to know what is the current population genetic architecture is. So this genetic architecture is a technical term, so which means two things, which encompasses mainly two things. The nucleotide changes, which are basically single nucleotide polymorphisms or big variations. What are these variations present in the genome of particular population, and what are their frequencies? So the genetic architecture, to know the genetic architecture, so basically the variation and their frequencies, you need to have population level data. So you need to uh, identify individuals in a population, sequence them, and use that sequence data to identify the variations in the genome and calculate the frequencies and check how these frequencies are differing between different populations, because that is actually a very important component. That is what makes all the populations of the world very unique to each other. Some are very similar, some are very unique. So the similarity and uniqueness is given by the presence of these variations, presence or absence of these variations, and if present in both the populations, what is the frequency difference of these variations, right? So that becomes very important. So for that, we have different yeah, so this is, I think, from 2010. 2010 happened for the first time. We had a global cohort of individuals of different ancestries sequenced together and checked for different types of variants. So that was the Thousand Genome Project. We discussed this last class as well. So yeah, population genomics involves the genome-wide study of variation within and among populations, because that is very important, right? So within population also there can be heterogeneity, among population also there can be heterogeneity, right? To infer demographic history, natural selection, and adaptation, and it can quantify the genetic diversity within and between populations again. So Thousand Genome Project is a very good example of that. So this project happened in 2010. There are, I, I think I have provided a few papers from before. Yeah, again, if you did not attend the earlier classes, so this is the Thousand Genome Project homepage. You can check out the data and the papers associated with it. Right. Okay. Okay. Now, what kind of information can you get from it? So, firstly, majorly broadly, you can talk about two types of information that you can get from this population level data. First is basically the ancestral information, meaning what kind of ancestry do these individuals come from, what is the proportion of different ancestral components in their DNA, in their genome. Firstly that, and the second thing is basically disease specificity, like any particular disease that is more frequent in that population compared to other populations, and so on. Right. So one such example is basically this lactase persistence allele, which is basically the lactose intolerance variation in Europeans, where you have positive selection of that particular variant in this LCT gene, LCT locus. So what happens is basically you have this particular variation in the genome of European individuals, but this has been going the variations has been there in the genome of Europeans for a very long time, and it has not been wiped out because of positive selection. And for this, a lot of Europeans uh, uh, are actually kind of intolerant to lactose, unlike Indians. So that is a very good example of that. Uh, Again, I talked about population structure and admixture, basically the ancestral component to it. What are the ancestral components uh, of an individual genome, right? So for example, uh, in India, this is a very good example. So we have different ethnicity people living in India. So India has documented around 4,600 ethnicities present. Some are tribal, some are non-tribal. But the major two populations that you can see are basically an admixed population of ANI. ANI is ancestral North Indian, ASI is ancestral South Indian. So we have, in terms of genetic architecture, we have two major populations in India, and there are smaller populations as well, but these are the major ones. Now, uh, for due to this specific nature of the environment that one individual is interacting with or a population is interacting with, so it can take up, sorry, so it can take up different adaptations. So, for example, in Tibeto-Burmans or Tibetans, basically we have uh, high altitude adaptations of some particular genes. So also there will be certain genes which help in keeping. So, for example, a person staying in Antarctica or a population staying in Antarctica would have certain positive selection of certain genes or certain variants that can help them keep out the cold, right? So those kind of adaptations we can study from this population level data. So yeah, this is a figure from the 1 KGP publication again. So these were all the places from which they collected the data. Again, you see there are like so five populations, I think. So this is, I think, Gujarati. This is Bengalis. This is Punjabis. I forgot the other ethnicities. So basically, this covered the entire world and collected around 1,000 samples, where they combined the data and analyzed what are the similarities, what are the difference between these populations. So on the below plot is, if I'm not wrong, so this are the total number of variants in a population. That this is a distribution. So these are in millions. So on an average of an individual, we have around 4 to 5 million variants. But you see some populations having lower number of variants per genome. Some are having higher number of variants. So there are systematic differences between these populations, right? And if I'm not wrong, this is the number of singletons. The singletons are basically variants that are found only once in the whole population cohort. So that also is very different for different populations. So although we are very similar, all of us together, but we have our differences. We have our differences in terms of genetic architecture. So that goes for the ancestral part. Again, ancestry is defined in terms of, like, some ancestral populations, they have come and admixed together, and they have created a new population, or that population is an admixed population of older populations, right? So that is what is given by these admixture plots. So this is what is known as admixture plots. So for example, you take one such example. So let's see this one, CE. you, right? This is one particular population. What the bar shows is basically the dark blue is a component of one particular ancestry, and the light blue is component of another particular ancestry. So this is a good admixture between two different admixed populations. Again, if you compare to this CDX population, this is actually one ancestry predominantly high and others are very low. If you see a darker green slightly here. So the populations are very different in terms of the admixture and what kind of admixture has taken place. But other than that, you can also reconstruct effective population sizes through time. You can model this using certain statistical techniques. One of them is basically PSMC, pairwise sequentially Markovian coalescence, and SMC++. SMC++ is a very well-known technique to do that. So this is, I think, from the 1 kg to 1,000 genome publication as well. So they reconstructed the effective population sizes of different populations, and you see there are distinct differences between different ancestries. So this populations come from a similar type of ancestry. The red ones come from a similar ones. The green ones come from similar ones as well. So there, at this point of time, there was some population bottleneck, and after that, the population started rising again. So that particular bottleneck is what is known as the out of Africa bottleneck, which is basically the idea that non-African populations show a strong reduction in effective population size around this region, which is like 50 to 60 thousand years ago. So that is also very important. And in Europeans, we observe more stronger and more prolonged bottlenecks compared to Africans and reduced genetic diversity. So if you compare to African population, European populations are much less diverse in nature, and they had much more prolonged bottlenecks and reduction in population compared to Africans. And if you consider archaic admixtures, so if you look at the ancestry in terms of archaic ancestry in terms of like Neanderthals and so on. So this revealed that Neanderthal integration into non-Africans and the Denisovian admixture in Oceanians. So we found a lot of this, yeah, historical evidences through sequencing. So that is, I think, really fascinating. Again, so as 10,000 years ago, humanity started doing agriculture, and from that moment, we see an explosion in population growth in terms of genetic diversity as well. So that is also very important. Okay, so we were talking about ancient genomes. So let's come to ancient genomes. What are ancient genomes? So the first ancient genome is basically that was sequenced from a hair sample, which is this Paleo-Eskimo individual of the Sakak culture. This was done in 2010, and this individual was 4,000 years old, and this fossil was the DNA was found in Greenland. So the thing is, since DNA in many cases for ancient humans and for ancient individuals, even if you find the remains, the DNA is not usable, or the DNA has been destroyed or something like that. So it's very hard to find high quality genomes of ancient individuals, but this was one that was found in 2010 and sequenced around 20x coverage, covering about 79%. So the main finding was that this particular individuals coming from this particular group, they were closer to Siberian populations rather than to modern Inuit or Native American population, right? So and it also provided migration patterns. So what kind? So if you can find an individual, an ancient individual, and find ancestral information of that individual in terms of what are the components of the ancestry of these individuals' DNA. So if you can do that, you can have an idea about which two populations actually got admixed and what can be the migration patterns of these individuals through different populations that came together and admixed, right? So that is also very fascinating, I think. So yeah, also we identified different phenotypic traits like brown eyes, thick hair, dry earwax allele, and other things as well through sequencing, right? So that also is very fascinating. And so another example is basically sequencing of a Neanderthal genome. So this was gotten from Siberia, and this genome was extracted from the toe phalanx, and and it confirmed that Neanderthals contributed 2% of DNA to non-African humans as well. So again, this paper is very fascinating according to me, and if you have interest, please take a look. I think this is a 2014 paper, but this is a fascinating finding. Okay, the other was the Denisovan genome, which basically was gotten from a finger bone in, of a, in a cave in Siberia, and it contributed to 5% of uh, uh, Melanesians and Aboriginal Australians. Right. So also we can get different interbreeding evidences. So it can reveal multiple integration events between modern humans, Neanderthals, and Denisovans, and this can help us understand the complex spatio-temporal population interaction. So again, I think from such genetic information, you can derive a lot of information in terms of their ancestry, in terms of their disease specificity, in terms of their migration patterns and everything. So, and also admixture with ancient human beings. So that is also, I think, very uh, interesting. Okay, so we talked a little bit about whole genome duplication before population genetics, but I just come back to that again. So again, genome duplication is the process, as we discussed, by which a region of the DNA coding for a gene creates additional copy of the gene, right? So generally, we humans are diploids, so we have two copies of the same gene or the same DNA and same chromosome. Haploid is basically half of the DNA content of that, so single copies of each of the DNA molecule. So our reproductive cells, like the sperm cells and the egg cells, they contain uh, haploid DNA content, but the rest of our somatic cells, other than the germ cells, the somatic cells contain diploid diploid DNA or diploid chromosomes, which is 2N. Similar to gene duplication, whole genome duplication is a process by which an organism's entire genetic information is copied once or multiple times, which is known as polyploidy. So these are in modern cases, when we see polyploidy, it is generally associated with some disease. So, for example, if we see Down syndrome, which is the trisomy of chromosome 21, so it becomes an extra copy of chromosome 21 is present, so that is also an example of polyploidy, but that leads to disease, right? So we are talking polyploidy and whole genome duplication in terms of evolution and natural selection. In some cases, this may provide evolutionary benefit to the organism by supplying it with multiple copies of a gene, thus creating greater possibility of functional and selectively favored genes. So the point is, if the important genes, the genes that are important for your survival and reproduction, if those are copied multiple times, if those are replicated multiple times, if those genes are affected by certain variations that can nullify the uh, genes function, basically turn them into pseudogenes, you have other copies of the same gene to be which can take its place, right? So that becomes very important. So evolution-wise, these polyploidy events lead to massive cellular changes, including again doubling of genetic materials, changing in gene expression, increased cell size. Gene loss during this diploidization is not completely random, but heavily selected. Genes from large gene families are duplicated. On the other hand, individual genes are not duplicated. And overall, these things can have both short-term and long-term evolutionary effects on an organism's fitness in the natural environment. And also, uh, uh, like if it does not cause a disease, this is actually very helpful. But if it causes a disease like Down syndrome, this is actually not great. It'll be negatively selected. Genome doubling provides organisms with redundant alleles that can evolve freely with little selection pressure. As I was saying, if you have multiple copies of the same gene, if one gene gets mutated or truncated or knocked out, you wouldn't mind because there are other copies of the same gene to take up its function. The duplicated genes can undergo neofunctionalization or subfunctionalization, as we talked about earlier, which could help the organism adapt to new environment or survive different stress conditions. But one important thing that comes out of this is speciation. So sympatric speciation can begin with chromosomal error during meiosis or the formation of hybrid individuals with too many chromosomes, such as polyploidy, which can occur during whole genome replication. We have identified different types of polyploidy that can lead to reproductive isolation of an individual in the polyploid state. In some cases, polyploid individuals will have two or more complete sets of chromosomes from its own species in a condition called autopolyploidy. The other form of polyploidy can occur when individuals of two different species reproduce to form a viable offspring, also known as allopolyploid, which is basically meaning uh, other polyploid, right? So yeah, so it occurs where gametes from two different species combine. So you'll have different set of DNA content, and you combine them to form a new organism. It has been suggested that polyploidization even creates new species via gain of adaptive traits or by sexual incompatibility with their diploid counterparts. An example would be recent speciation of allopolyploid spartina or S. angelica, which is a polyploid plant which is so successful that it is listed as an invasive species in many regions. So this, from an evolutionary perspective, it is very like kind of advantage in a very advantageous position. Okay, now uh, we come to something known as the two-hour hypothesis, which is basically uh, the hypothesis that in terms of whole genome duplication, uh, it differs. Uh, so there's this person which is the sorry, yeah. So yeah, so there's this person called Ono. So he hypothesized that at a certain point in the evolutionary history, the genomes got duplicated. So it happened twice. So it got duplicated, and through whole genome duplication, as we talked about, new species can be formed. So around this time, which is around like 500 million years ago, so here the duplication occurred, where basically you divided the fish into amphibians and cartilaginous fishes. So from fish we developed into a little bit into reptiles and amphibians. So that is the theory behind the two-hour hypothesis. But now it has been identified that the two-hour hypothesis basically distinguishes between jawless and the jawed vertebrates. But now this theory has been slightly disproved, and we have gotten like 3R kind of it happened three times this particular speciation event. So there are papers regarding that. So this picture is from one such paper. Uh, so again, if you once sequencing of this elephant shark genome, it had showed that there are four Hox clusters like mammals, indicating that 1R and 2R preceded the radiation of jawed vertebrates. So yeah, the idea is that you had whole genome duplications, and from that speciation happened, new species formation. Okay, so what can happen if genes are duplicated? What are the fates of these gene duplications, right? So first example would be of a singleton. So singleton is basically a single copy of the gene. Starting from a singleton state, a gene can become duplicated, then present in two copies via tandem duplication. It's just the same region of the genome has been duplicated twice. So this is one copy. This is the normal gene. Now the gene has become two copies, right? Now hypofunctionalization. So a gene has become duplicated, being present in two copies via tandem duplication, transport duplication, or whole genome duplication. Right? Now, hypofunctionalization involves reduction in expression of both the copies of a duplicate pair through a threshold level at which both copies are needed for specific function of the gene. So the idea is that you have the same gene now in two copies. What you do is you reduce the expression of both the genes, which is this hypofunctionalization, and so that both the genes can be both the copies can be expressed to reach a certain threshold of the gene expression which is required for the cell to perform, cell to function, right? So both the copies will be maintained. Subfunctionalization would be basically the division of functions such that two members of a pair confer only part functions of the progenitor singleton. So basically you have, instead of singleton, you have now two copies. So what you do is, say for example, this protein you usually earlier used to, sorry, this gene used to code for some important protein, right? So now it has two copies of the same gene. So what it now is doing is, since the protein has a lot of subunits, so let's assume histone has 8 subunits, right? So for example, if that happened, so half of the subunits would be produced by one copy, the other half will be produced by other copy. So that is what subfunctionalization is, right? So another type that can happen is new functionalization, which is basically one member of the duplicate pair can be assigned a new function. Dosage balance can be used to mediate detention of specific classes of genes that are dosage sensitive to maintain their stoichiometric relationship with partners in multi-component interactions. So basically, again, so if you have a lot of copies, you kind of silence one of the copy. So dosage compensation can happen. Uh, so these are different types of uh, ways the genome evolves to accommodate these extra copies of the genome, right? Okay. Balance can mediate retention of specific classes of genes that are dosage sensitive to maintain the stoichiometric relationship with partners in multi-component interactions. Nevertheless, the compensatory drift refers to the observation that duplicates can diverge, but depending on the total expression, a dosage balance effect will still operate in the partner. So it is, the compensatory drift and hypofunctionalization are kind of similar in terms of they reduce the dosage, they reduce the expression of the genes, but in compensatory drift, what it will do, it is reduce expression of one copy of the gene very low and the other copy of the gene very high. So it kind of balances out, right? So again, and ultimately, it can be a neutral variation. So it can be a neutral variation, which is basically the different members of the duplicate pair might have different expression levels under certain circumstances in which the dosage or function of an object is not an object of selection. Similarly, to apparent neutral variations of singleton genes. So basically, it doesn't matter if that gene is duplicated because the dosage of the gene does not matter. It has to be expressed. If, however, it is expressed, it does not matter. So in those cases, that component is known as neutral variation. So, what is, after studying all this, what is actually natural selection? Then the idea of natural selection is that you have, so through randomness, these mutations and these variations in the genome are introduced, but not all these variations will be useful, right? Some will be harmful, some will cause disease, some will cause deaths, and some will be very important and very efficient in terms of survival of the organisms and reproduction of the organism, right? So the natural selection is the differential survival and reproduction of individuals due to differences in phenotype. It is a key mechanism of evolution, the change of heritable traits characteristic of a population over generations. And Charles Darwin famously used this term natural selection, contrasting with artificial selection, which is intentional, whereas natural selection is not. So the idea is that if you gather more beneficial mutations and which is helping you survive and reproduce, you as a genome content, you will be selected for propagating into next generations because you are more fit to to survive, right? So survival of the fittest. So again, so this kind of this no graph shows basically some mutations which are not acceptable, some mutations which are acceptable, and they grow into next generation. So yeah. Okay, now how do you statistically identify if there are variations that are going on in the genome, and how if selection is happening or not to these variations? So there are a couple of ways. One is the codon or the gene-based ways, and the other is basically allele frequency-based. So we'll talk about the gene-based, and this is the best known characteristic for detecting selection, which is Ka by Ks or Dn by Ds, which is very well known, which is basically the ratio of non-synonymous mutations to synonymous mutations in a gene. The idea is that non-synonymous mutations can change your amino acid content and ultimately the protein structure. So it will have a very direct impact on the expression of the gene, but synonymous mutations do not have that, right? So these mutations are kind of neutral, so they do not actually uh, confer to any differences in gene expression or protein production in the cell. So that is what we compared, dn by ds, which is the ratio of non-synonymous to synonymous mutations. The statistic compares the rate of non-synonymous mutations per site with the rate of synonymous mutations per site. Because synonymous mutations are assumed to be functionally neutral, their substitution rate provides a baseline against which the rate of amino acid alterations can be interpreted. A relative excess of non-synonymous substitutions indicate ongoing positive selection favoring novel protein structures, on, or else cessation of negative selection against protein alterations. Uh, so if you have a greater than one value for Kn by Ks, which is you're having higher number of non-synonymous mutations compared to synonymous mutations, which means the non-synonymous mutations are under selection, positive selection, because they are forming new proteins which are actually beneficial. So those mutations are still there; they have not been selected out, right? So if it is greater than one, it is basically it is acting as positive selection. If it is less than one or smaller values, they indicate ongoing negative selection against deleterious mutations and consequent preservation of protein structure. These methods may also be applied across an entire open reading frame or some subdivision thereof, down to an individual codon, as different regions of the protein may be subjected to different selection pressures depending on the function of the protein, right? Various models for calculating synonymous and non-synonymous substitution rates take into account different probabilities of different mutations. So transition and transversion. If I'm not wrong, transition is basically purine base replaced by purine base. Transversion is the purine replaced by pyrimidine or something like that. I have forgotten. Just check it out. But yeah, the idea is that some mutations are more frequent in nature compared to others. Right. So the concept of neutral theory of molecular evolution is that the genetic change is attributable to genetic drift rather than Darwinian selection. So uh, you need to understand the difference between neutral and adaptive change in the genome. So how do you do that? Generally through statistics like Kn by Ks or Dn by Ds, or you have other statistical techniques like Tajima's D, or using allele frequency differences across different populations. You can get an understanding of that. Yeah, so neutral theory enabled the development of tests for selection by assisting sophistication of models of genetic drift. In many tests for selection, which are neutrality tests, researchers compared empirical data against data generated by simulations of drift, which act serve as null hypothesis. So basically, one hand you have your population level data, on the other hand you can simulate uh, the mm, uh, the population level data across different selection uh, events across multiple, so millions of years, and you can simulate a data for that, and then you can compare against both, and this will act as a null hypothesis, right? So other neutrality tests may use background rates of change inferred from whole genome analysis to furnish a null hypothesis. So again, this plot actually shows different methods for detecting selection at macroevolutionary level, traits that are conserved across many clades of a phylogeny, but that showed extreme differentiation in one or few lineages are likely candidates for selection. So again, so if you have this is your ancestral sequence and this is your current sequence. So if you compare it, you'll have some non-synonymous and synonymous mutations, and then you calculate this Ka by Ks. If it is greater than one, that means your mutations are against uh, positive selection. Yeah. So metrics such as Ka by Ks compare the rate of non-synonymous substitutions in a lineage to the rate of synonymous substitutions, which are assumed to be selectively neutral. The McDonald-Kriegman test, or the NKT, we'll talk about this, and Hudson-Kriegman test, we will not talk about this, hinge on the intuition that the levels of interspecies divergence and of interspecies polymorphism are governed by mutation rate and correlated unless selection of something is happening at the population level. So what does that mean? So again, I come to these tests. One of them is the MK test, McDonald treatment. So the idea of this test, yeah. So the idea of this test is to detect natural selection acting on protein coding genes by comparing it within species and between species. How different, what are the variations that are present within species and what are the variations that are present between species. So the idea is, the concept is that neutral theory predicts that rate of non-synonymous to synonymous should be the same within a species and between species. So the ratio should be fixed for within species and also fixed at a fixed difference with other species. Deviations from this, if there are differences between these, which that can indicate selection, some sort of selection is going on. So these are the terms that are used. So for synonymous within species, it is used as PS. For non-synonymous within species, it is used as PN. Again, for other species, it is used as divergence between species as DS and DN. So the interpretation is, under neutral evolution, PN by PS is approximately equal to DN by DS. So across species, the the rate of non-synonymous mutations to compared to the rate of synonymous mutations com should be comparable across different species. If positive selection is going on in a particular species, their DN by DS will be higher, significantly higher than other species. And if purifying selection is happening, then within species differences will be higher than between species differences. So, which means excess non-synonymous polymorphisms which are removed before fixation. So, this is the test statistic that is used for this particular test, and it is given by 1 minus Pn by Ps by Dn by Ds. So, this is the alpha. So, alpha greater than 0 is evidence for adaptive evolution. Equal to 0 tells about neutrality, and if it is negative or less than 0, it tells about strong purifying selection. Right? Okay, I thought somebody. Okay, so yeah, we come to the other test, which is the Tajima's D test. Sir has also covered this. So here also the idea is to find deviations from neutral evolution by comparing two measures of genetic diversity. So there are a couple of elements here. First is the pi, which is basically the average pairwise nucleotide differences. The second is the theta for Waterman theta, for theta w, which is basically expected diversity from from the number of segregating sites. And your Tajima's d is given by pi minus theta w divided by the root over variance of pi minus theta w. Right. So again, Similar concept, if it is equal to 0, close to 0, neutral evolution. Less than 0, excess of rare alleles, which means population expansion, purifying selection. And if it is greater than 0, it means excess of intermediate sequence alleles, balancing selection or population bottleneck. So here is an example of that. So if you take these four sequences, so there are six variable positions in this. So I think I can point out one. Uh, I think it is two. Basically, there are six. If you count, there will be six, three, four. Uh, I'm missing some here. So that there are six of these variable positions across the sequences. So the theta W, which is the Waterson's estimator, is given by this formula, which is S by AN, where AN is a summation of 1 to n minus 1, 1 by I. So in this case, there are four sequences. N is the number of sequences. So there are four sequences. The theta W is calculated by so first you have to calculate an. So an will be 1 by 1 plus 1 by 2 plus 1 by 3 because it goes to n minus 1. So that becomes around 1.833. And the theta w is given by s by an. So s is here 6, an is 1.83, which is 3.27. So theta W has been calculated. Now you calculate pi. Pi is basically the pairwise differences across six pairs. So sequence 1 versus 2, 3, 4. Sequence 2 versus 1, 2, 3. Sequence 3 versus 1, 2, 4, like that. So pairwise differences, there are total 18 differences if you count like that. So pi will be the the total number of pairwise differences divided by the total number of uh, variable positions in the sequences. So it is 18 by 3, 18 by 6, which is 3. And the numerator of this, I'm not calculating the variance. And the numerator of this is negative. So 3 minus 3.27, pi is 3, theta w is 3.27. So it becomes negative, negative 0.77, which means there's an excess of rare alleles because if you calculate variation and basically divide it, it will still be negative because this term is going to be positive. So I've not calculated that for simplicity. So in this case, the d is less than 0, which means excess of rare alleles, which is population expansion and purifying selection. So yeah, that is all in terms of the content of this week's. Now we'll discuss the assignment questions. Before that, does anyone have any questions? Okay, one thing Sir has asked me to mention that please do read the papers that he cited and also like the ones that I have given you, so that you can get a better understanding of how this kind of research is conducted, how this kind of work is done. So you'll have a better understanding if you read the papers, and also like if you are in this field, reading papers like these from Nature Genetics is going to be very helpful for your uh, kind of growth in terms of understanding the science as well. So please do that. Okay, we'll start with the questions. I'll just be back in a minute. Okay, so both of these species, X levis and X tropicalis, are basically two different types of frogs, right? So these are used as model organisms in labs. And this X tropicalis have a shorter uh, generation time and they have a diploid genome, and this X levis have a tetraploid genome. So we have any answers. Okay, the idea is that it has a larger size than X tropicalis, so the statement is true. Uh, and the second statement is X levis has one excess round of whole genome duplication compared to X tropicalis, which is basically it's tetraploid compared to diploid here. But the size of the organism is not related to the whole genome duplication. So statement one and two are correct, but statement two is not the correct explanation for statement one. So yeah, these two options should be looking at. Okay, moving on. If you have any doubts or any questions, please write in the chat box or feel free to mute yourself and ask. Okay, so these are like factual questions. There are a lot of factual questions in today's assignment. So yeah, okay. Please try to answer. The answer is six. So yeah, there's a very good history of banana, right? So think plus two. Sorry, seed. This is a very important. So this is a very important in terms of agriculture, but very interesting in terms of the genomics. So how did this actually happen? So again, if you're interested, please do give it a read. Okay. Here the answer is basically C. You are correct. No, sorry, B. I think you said C. Yeah, C is not correct. B is correct. So, first option, most evolutionary variations at molecular level happen to increase fitness, which is not true. In some cases, it can have deleterious, in most cases, it will have deleterious effects. In some cases, it will increase fitness, but like mostly it will be neutral, right? So neutral is the maximum proportion of these variants. So first option is wrong. The second option is actually correct. Most evolutionary variations happen at molecular level are caused by random genetic drift. It is a very random process. So these are not targeted. The mutations are not targeted at all. They are random processes. But if by those random processes, the mutations turn out to be good for the species, it stays in the population through positive selection. But yeah, C is also not correct because most evolutionary variations at molecular levels are caused by population positive selection. That is not true. They are caused due to randomly accumulating these mutations. These are not caused by uh, positive selection. Okay, it is caused by random drift. It's a purely random process. Positive selection will happen after the mutations has occurred. So at the molecular level, when the mutations occur, after the mutations has occurred, then the selection will be happening. Positive selection will not induce these mutations. So the third option is not correct. Second is correct. Right. Here the answer is actually none of these. The second option is kind of true, but it's not fully true because it has not fused from head to tail. The chromosome 2A and 2B have fused, but it has not fused from head to tail. There are breakpoints in both the chromosomes that have fused. So partly 2A and 2B has fused to give us the chromosome 2 uh, uh, to form humans. So basically, uh, the chromosome 2A and 2B in chimps have been partly fused to form the chromosome 2 in humans. So none of these options are correct again. Okay, moving on. That's correct. These are factual questions you need, sorry, these are factual questions you need to know the answers to, but the yeah, correct answer is here. Wait another minute. No. So try to understand what is dn by ds. Dn by ds is the rate of non-synonymous mutations by the rate of synonymous mutations. So in general, when there is no positive selection happening against certain genes or certain proteins, what will happen? You'll have more accumulation of synonymous variations compared to non-synonymous, because mostly these variations should be neutral and very few of them would actually be contributing to the change of the amino acid. So the answer is dn by ds less than 1. So your synonymous mutations should be higher compared to your non-synonymous mutations in any general condition, right? If it is greater than 1, that means it is under some selection. Then you have more number of variants that are changing amino acids compared to your neutral variation. That should not happen in a general condition, right? Only when some selection is happening, then that will happen. So in a general condition, most of the genes will have dn by ds less than 1. The DSS will be much higher than the synonymous will be much higher than the non-synonymous variations. Right? Okay. Okay. So firstly, what is selective sweep? Let me explain that. So selective sweep is basically you have a very beneficial mutation that has occurred in the genome. So what it selective sweep is basically in the further generations, that particular variation frequency increases rapidly. So because that is a beneficial mutation, positive selection on that occurs. So if negative selection, let's go with the options now. If negative selection is happening, it means that the deleterious mutations in the genome, their frequency are getting lowered. So they are negatively selected, so they are getting weeded out of the genome. So the first option is actually correct. Negative selection will have excess of low frequency alleles, right? Second option, positive selection will have excess of high frequency allele. That is not true because during if positive selection is occurring, some variants will have the frequencies increased or be in the population more. But that doesn't mean you have very high frequency alleles. So those positive selection, and there's a difference between positive selection and selective sweep, right? Positive selection is basically selection of that variant to be propagated to the next generation, right? For selective sweep is selection of that variant, and in the upcoming generations, the frequency becomes very high. So the second option is actually not correct, but selective sweep will have a high excess of high frequency alleles. Selective sweep will also have an excess of low frequency alleles, right? Because uh, you from both ends of the spectrum, selective sweep will have high frequencies. Like you have high frequency alleles from selective sweep, you'll also have low frequency allele from selective sweep because those alleles are still undergoing positive selection. So negative selection, the first option is correct. Negative selection will have low frequency allele in excess. Selective sweep will have both and the both of the spectrum. So some mutations will be like taken and very high, the frequency will reach very high numbers, and some mutations will be present at the lower end, and selective after selective sweep has happened, those will also be uh, move to higher frequencies. So, except for the second option, the rest of them are correct. The last option is a little bit tricky in terms of understanding, but you should get the first and third option, right? The last option is also actually true. If you read about selective sweep, the low frequency alleles also get accumulated. So, apart from the second option, all the options are correct. So, please take a look and to look at the question properly and try to understand what it is saying uh, before you mark the answers. Okay, I hope this is fine. I'm again repeating. So in selective sweep, some variations will have these higher frequencies because those are very beneficial mutations, and it will lead to the surrounding regions having lower uh, number of variations of that gene, if you say. So at the higher end, you'll have a lot of alleles in terms of frequency, and the lower end also you'll have a lot of alleles frequency. Yeah. Okay. Okay, to answer this question, let's go back to the slide where I discussed. So this is the NK test, right? So you have these two. Within species, you have PS and PN, synonymous, non-synonymous, and between species, you have DS and DN. So the idea is, if you have positive selection between species, DN by DS should be greater than within species, PN by PS. So basically, uh, excess non-synonymous fixed differences, and if you have purifying selection, you have the opposite, which is PN by PS greater than DN by DS. So now let's go back here. The answer is the, sec the first option, sorry, the first option, which is DS by DN greater than PS by PN. So within species, DS by DN is greater compared to inside species, right? So these are conditions from these tests. So okay, let's move on. This is a straightforward question. So the answer is actually all of these, but all the options are given as correct. So segmental duplication can happen, local chromosome trisomy can happen, whole genome duplication can happen. Anything of the three can happen to form this long regions in the genome. So duplication is the major thing. All any type of duplication is possible. So okay, let's move on to the next one. answers all of this. Pseudogenization can also happen. If you remember, if you have multiple copies of the same gene, and what happens is basically all the copies are duplicated at the same point of time. So ultimately, none of the copies are functional. So that can also happen. So gene duplication. So if the duplication is happening, say for example, in a whole in a part of the gene where the promoter is, or in a part of the gene where it is an important component where something can bind to that gene. So if that regions are duplicated and those kind of things happen, so the gene function can also go off. So pseudogenization can also happen. So all of this. So sub-functionalization and new functionalizations are common. So you have some, if you have duplicated genes, one gene will take up one function, the other gene will take up the complementary function. New functionalization when the other copy will take up a new function altogether, but pseudogenization can also happen. So all of these is the answer. So like these kind of questions, they like kind of try to trick you. So yeah, try to read the questions more carefully. No, it's not going to be a homogeneous distribution. And I think you chose I think you chose A and D. I think the answers are B and C. It's not going to be a homogeneous distribution because one round of genome duplication, the rate of synonymous substitution, it's never going to be homogeneous because it's always random. So it is always going to be heterogeneous. So homogeneous distribution will never find something of a homogeneous distribution in genomics. You will very rarely find because everything, these mutation rates are very random in nature. So this after the mutation occurs, it will always be a heterogeneous distribution, and it'll have two peaks basically. So after one round of duplication, it can have two sets of kind of peaks. So I am also not very sure about the explanation to this, so I have to kind of look it up. But yeah, this is the correct answer here. So yes, let me check, do a quick check. Yeah, so it can be a bimodal or a multimodal distribution. So mainly a bimodal distribution that is why it will have two peaks, uh, and because it will have kind of like basically two major values of frequency around which it will cluster more. So yeah, it will be bimodal and it will be heterogeneous. Homogeneous, nothing will happen. So the answer is basically B and C, not A and D. Right, I think I have found a paper if you want to take a look. One second. Yeah, I think this paper kind of explains that uh, very well. Yeah, you see, you have like this multimodal positions, but like mostly it will be two in one. If it is undergoing one segmented duplication, you will not have these multimodal peaks. You'll have a single peak. So, sorry, bimodal peak, not a multimodal peak. Yeah, let me see if I can find anything better. Yeah, but the idea is that after one set of um, duplications, it will have two peaks and it will not be a no homogeneous distribution. After multiple rounds of whole genome duplication, it will have multiple peaks because it will like multiple copies of the genome. So one, if you have one copy of the genome, you have a single peak. If you have two copies, you have like a bimodal thing. If it is multiple copies, you'll have multiple such peaks. So since it is gone for one round of whole genome duplication, you'll have kind of two peaks. So I hope this was clear. Yeah, the answer is all three. I think this was self-explanatory. So yeah, increased adaptability will increase genomic diversity and also can lead to speciation as we see into our hypothesis. So okay, this we have not discussed. FST. FST is basically It stands for statistics. Basically, allele frequency differences across population. I think this is the code for it. Yeah. So different populations, what are the different, so same variant, what are the allele frequency differences? That is how FST is quantified. So we have not discussed this, but this is a very important metric to quantify differences in population structure across different populations. Okay. Yeah, I'll not dwell on this question, but the answer is 15%, the average FST between human populations. So average around on an average, we have 15% different in terms of allele frequencies of the between any two random populations. Okay, let's move on. Uh, the second is also present. The correct answer is A, B, and D. Uh, so it is basically, if I'm not wrong, it is yeah, likelihood, single likelihood ancestor counting. So this is another technique by which you can calculate this. So I don't think this is the single likelihood. Yeah. Moving on. Answer is actually all of them, both A, B, and C, and the final option is A and B together. So, mutation with heterogeneity, GC bias, gene conversion, all of these can contribute to uh, false positive selection in codon-based tests. So, basically, if you have a GC bias somehow incorporated in while gene duplication, gene conversion, or in during DNA GC content increase in DNA due to gene conversion and recombination. This will give you, since you're doing codon-based tests, anything overrepresentation, in this case, overrepresentation of GC can give you false positives. So all of the options would be correct. So yeah, the important thing is this is a codon-based test, and if you have a codon-based test, any type of biasing will give you false positive. I hope you're calculating. Okay. Uh, okay. Okay. So you have you have these four sequences. So if you do pairwise differences between these individuals, so there will be, I think, if I'm not wrong, so if you have to calculate what are the differences between sequence, right? So sequence one versus two, three, four, sequence two versus one, two, three, one, three, four, sequence three versus one, two, four, and four versus three, two, one. So you have to find the total number of differences, and then you have to weigh it by the total number of individuals. So sequence one versus two, three, four, when you do it, you multiply it by nine. Sequence two, when you're considering, you multiply it by five. Sequence three, when you're calculating, you multiply it by four. Sequence four, when you're doing, multiply it by two. So when you do that, you'll find the total number of pairwise differences is coming as 537. I've not done the individual calculation here, but yeah, the total number should come around 537. So it is a tedious thing to do, but the idea is when you compare sequence one versus the rest of them, You weigh it by the total number of individuals. Same with sequence 2, sequence 3, sequence 4, etc. So that comes as 537. Now the total number of pairs, how many pairs are present? It's basically 20 times 19, so 20 individuals are present, right? So 9 plus 5 is 14, 14 plus 4, 18, and 20, right? 220. So 20 individuals present. We're comparing 20 individuals to the rest of the 19, so the total number of pairs will be 380. 20 times 19, and if you calculate the sequence length, this is around 36. So the pi is given by the weighted differences divided by the total number of ordered pairs times the sequencing length, which comes as approximately 0.390399. So I'll quickly go back to the formula here. Yeah, sorry. Yeah. So the pi was total number of differences by the total number of basically positions of difference, as in like how many position in the genome there are differences, and the numerator will be the total number of differences. Same thing we are doing here. Okay. Yeah. So the total number of differences, in this case, it is weighted because of there are certain number of individuals. So that comes to 537. And total number of pairs will come as 380. The number of the number of combinations you're doing, and the sequence lengths come as 36. So if you do this, the answer will be 0.0399. This is a kind of a tedious one to do. I tried it. This took me a long time. So yeah, this is a tedious thing to do, but the idea is simple. You just put the things in formula. Just you have to keep in mind that you have to, since you have these several individuals having these sequences, you need to when you're calculating the differences, you need to weight it by multiplying it by the total number of individuals for that sequence. So yeah, this is around 0.0399. Is the explanation okay? Yeah, yeah, that is correct. Pairwise, you need to calculate and then weigh it by the total number of individuals. 1 versus 2 plus 1 versus 3 plus 1 versus 4. Yeah, times 9. Correct. This is just a tedious thing to do, but it's not that difficult. Okay. So next is the calculation of theta. Theta is basically going to be S by A1. S is basically the total number of uh, uh, variant sites here. It is, I think, 6 here. I think if you calculate, the total number of variant site is going to be 6. So 6 divided by the AN. So if you remember AN, let me go back. Yeah. So theta is S by AN. S is the total number of sites, and N is the total number of sequence. Right. So AN you calculate 1 by I equal to 1 to N minus 1. So here it will be 6 sequences. Sorry, 4 sequences. It will be the same 1 plus half plus 1/3, which will be 1.833. And there are 6 differences, 6 variant sites. Sorry. Yeah. The 6 variant sites here. So 6 divided by 1.833, which comes as like around 3.278, which is the second option. Right? So these are like basically formulas. You need to put the values and get the answers, but to put the values, you need to calculate these different things. So if I'm not wrong, here there are 6 variant sites. You have to see where those variant sites are, but yeah. Okay. Yeah. Okay. And finally, if you want to find Tajima's D, again, going back to the formula, if you want to find the D is basically pi minus theta w divided by variance of pi minus theta w. You have found pi and theta w. You have to find the variance across the 4 different sequences. And take the root over that. So if you do that, it comes to around, I think, negative 2.544. So just plug in the values in the formula, you will get the answer. I think it won't be an issue. Yeah. Just keep in mind while calculating the pi and the theta, so not the theta, while calculating the pi, sorry, the average pairwise nucleotide difference should be weighted by the total number of individuals for that particular sequence. So keep that in mind. Okay. Yeah. Pi minus theta divided by root over variance of pi minus theta. That is the uh, formula for Tajima's D. Okay. Yeah. So I'll not take time in these. So bar-seq is basically, I also am not very familiar with bar-seq, so uh, I'll not dwell on this. Found a paper, I think a few. I think they studied. Ah, I think we can take a look at this particular paper. The idea is that it'll give you basically barcodes. It will have a relative abundance of barcodes to give fitness to the individual sequences. And in evolutionary experiments, this bar-seq will save time and money, which will be required for a whole genome sequence. So basically, it's smaller sequences of the genome where you can tag with a barcode, and you can compare multiple of these sequences at one point of time. You do not need to do the whole genome sequencing. So bar-seq kind of helps with that. So that is the kind of the understanding that I have. I also do not have a great understanding of bar-seq. So yeah, I'll leave it at that. Okay, this is the final question. Yeah, sorry, I missed it. Yeah. So yeah, so during mutation accumulation evolution experiments, arbitrary fixation of mutations occur. So basically, arbitrarily, uh, these mutations are encompassed in the DNA, and then the experiments are conducted. But they are not like kind of fixed mutation. The fixed mutations you can do, but that would not be a good standpoint for an evolutionary experiment. For that, you need to have arbitrary fixation of mutations. So option C is correct. I think you gave the option D, if I'm not wrong. Yeah, so the other options basically frequent bottlenecks get uncontrollably introduced with time. So that is not the case. That will only happen if you are only giving in deleterious mutations. So if you give too much deleterious mutations over time, the organisms would not survive, and simulation wise also they are not supposed to survive. So frequent population bottlenecks cannot be introduced until and unless you do a specific type of mutations in certain genes which will cause this bottleneck. Other than that, uh, it won't happen. So the answer is only C, not A and C. Right. First option will be correct only if you do like not if the C option is incorrect, basically. If you do not do arbitrary fixation, if you do specific introduction of mutations which are deleterious in nature, then only the first option will happen, otherwise not. Okay, so yeah, that is all from my side for this week. Do you have any questions? Okay, then thank you for joining. I think, yeah, we'll have 4 more weeks left, so yeah, we'll continue. So, okay, see you next week, same time. Thank you for joining. Thank you.